So I think that's actually all I have. We are gonna have something a little special today. I think we're gonna have a live VR demo. So during that time, if you do have questions, we'll probably have to hold on to them until our speaker resurfaces from the VR demo. All right, so with that, I'll turn over to our speaker today. Please present Eric Bell, who is a grad student in DCMB.
All right. So thank you everyone for showing up. I'm actually defending in about a month, so I'm an old man of the department, and this might be my last time presenting in this room, and I've spent a lot of good times in this room. So anyway, I'll be talking about the parts of my PhD thesis that are done, basically meaning that the last portion is still ongoing research, but basically my PhD has centered around protein-protein interaction prediction and looking at the networks that come out of that kind of analysis. So why do we care so much about protein-protein interactions? Well, the reason is because they're kind of everywhere. At the center of function in a cell is the protein, and those proteins don't work by themselves. They need to be interacting with other things in the cell in order to confer their function. So if we want to understand what's going on functionally in a cell, we need to know what the network of protein-protein interactions are that are going on in a cell. And this can have a lot of therapeutic benefit, like if we want to design a drug for a protein interface, like mini binders or antibodies, or if we want to do protein design on those targets, or we want to figure out whether a point mutation will impact an interaction or something like that. We first need to know whether there's an interaction to be looked at in the first place. And so we have some experimental ways of doing this, but they generally have high error rates. You see a lot of complementarity between these sort of high throughput experimental data sets rather than coverage of the entire proteome. And so we can use computational approaches to supplement or refine these kinds of data sets until they are covering the entire proteome. And so now we'll talk about a program that I've developed for computational PPI prediction. And these are some shortcomings of the field that I wanted to address in developing that program. That first of all, most predictors focus solely on the idea of a functional interaction rather than a physical one. So to do those things I mentioned earlier of like drug targeting or point mutation interaction prediction, you actually need to know that the two chains are coming into physical contact. And a lot of the predictors that are on the market are only looking for things that have a functional association with each other. Another issue that I want to address is that a lot of programs, particularly machine and deep learning based methods, are only benchmarked in species-specific cases, meaning that if you just give it a protein, it won't know what to do with that. It needs to know that it's a protein from a human or from yeast. So the program I wanted to develop would basically just look at proteins as a whole rather than focus on single species. And then finally, a predictor that involves structural insight usually is too slow to be used in a proteome-wide context. So that's something that I really wanted to address in my program. So this is the program that I developed. It's called the PEPI pipeline. Basically, the main idea of it is that you have a pair of query sequences that go into a bunch of parallel modules that I'll be going into depth of what all those modules are, that each of them predict individually what the actual score is, whether the two are interacting or not. And then those scores get mixed together into one final prediction using a Bayesian consensus model. So the most important of these modules are based on the idea of interology, which is just basically a fancy way of saying, is there an existing interaction that looks like the queries that you put in? So essentially, we're saying if we have homology between A and X and B and Y, and we know X and Y are interacting, then we can probably guess that A and B are also interacting. So the easiest way that we can do this is just by sequence. So if we just sequence search through a database of high throughput experimental data, stuff that's been annotated with being a direct physical interaction or having evidence of being direct and physical. If we just search through that sequence database and get hits out of that, how sequence similar are we to existing interactions, or really even things that have been observed. And we do that just by splitting apart the query chains into 2 individual monomers, searching those monomerically through a database of all of the sequences of a bunch of dimers, and then pairing together the things that were dimers in the database to get our dimeric hits. And then we just score this one based off of the sequence identity, the harmonic mean of the 2 chains sequence identity. So we can get a little bit more complicated with this and go from just sequence identity, which is not very sensitive at detecting evolutionary relationships, to structure, which is very sensitive at detecting evolutionary relationships because structure is more conserved than sequence. So the way that we integrate this structural information is through a dimeric threading program called Spring, that was developed in the Youngjohn lab in 2013 or so. So I've reprogrammed it specifically for PPI prediction. And the way that it works is it's very similar to the previous approach, except instead of doing a monomeric blast search, we're going to be doing monomeric HH search, which is a threading program that tries to align the input sequences to structural templates. And then from those structural templates, we pull out our dimeric hits and then potentially even form a model of what the interaction should look like if the threading quality is high enough. And so I also have listed Spring and Spring neg here. There's sort of this module sort of serves double duty, that we have both a database of things that are interacting and also a false positive database that if we hit in that false positive database, it actually reduces the likelihood of interaction. That false positive database is constructed entirely of chains that are part of the same functional complex, but are not actually coming into physical contact with each other. So if we're hitting those structural pairs, we know that it's more likely to be a false positive due to functional association.
Can I ask a quick question?
Yeah.
Do you segment your chains into domains?
Yes, so that's something that's done similar to how it's done in, I want to say, earlier forms of iTaser, where it aligns templates to your query sequence. And if you see a section where there's one half that is aligned to a bunch of queries and another half that's aligned to a bunch of different queries, and then gaps filling the other 2 quadrants, then we'll split the chain into domains. So the way that we reconcile this, and it's only done specifically on the structural aspect of things, the way that we reconcile this is that when we split our query into individual domains, we then pair every domain of one sequence with every domain of the other sequence, and then just take the maximum score. And so the score that we're getting out of this is an internal scoring function for Spring. It's compiled of a bunch of different aspects of the threading process, and it's the quality of the model that you get out at the end. And most notably, it also includes an interface potential. So if the residues that are getting put together at the interface are not compatible with each other, that'll show up, as well as if it introduces steric clashing of any kind. And so for the things that are not homology based, we do incorporate a little bit of functional association insight, where we just basically look up whether the interaction is present in the string database. And if it is present in the string database, then we pull out a bunch of functional association features that are contained within, including co-expression, gene neighborhoods, gene fusion, gene co-occurrence. All of those are just raw scores that we pull from string and put it into the classifier at the end. And then finally, we sort of have a quick and dirty classification method that's based off of machine learning, kind of an old style of doing machine learning for PPI prediction. This is like circa 2010 or something like that. I believe this specific paper is 2007. So it's an old way of doing things, but it's quick and it's easy. So the way that we do it is we transform each monomeric sequence into a fixed length vector according to the conjoint triad criterion. This basically just counts up the frequency of specific sequence triplets in your sequence. It scans over the entire sequence and makes a frequency table of those triplets. That gets turned into a fixed length vector. You can concatenate the two fixed length vectors together for the two sequences and then put it through a neural network model. This is actually a shallow neural network model. It's just one layer of 1,000 nodes. And that's trained on the Spring database, the structured database that we had before. And so how do we actually get a prediction out of this now that we have a pile full of scores? The way that we do it is this Bayesian consensus model, which is based off of the naive Bayes criterion. So the way that we trained this consensus model is just by taking 800 interactions from the intact database, things that didn't necessarily have a structure, but we were highly confident that they were true interactions. And then 800 non-interactions from the negatome database. This is a database that is experimentally confirmed non-interactions. Getting non-interactions, like confirmed non-interactions for PPI prediction is really difficult, and a lot of people just sort of give up and randomly pair things and call the decoys negative. But that's not necessarily with high fidelity. So it's good that we have a database that confirms that these things are non-interactions. So for each of these 800 interacting and non-interacting pairs, we use the modules to score each pair. We then form a histogram out of those scores for each module. And then using kernel density estimation, we fit a curve to the histogram. And that curve is actually what we're going to be using for classification. That's sort of the statistical model that we've learned here. And so I have an example here on the right where the green histogram is negatives, the red histogram is positive, and then the curves associated with each of those two histograms are the actual model itself. This is specifically for Spring, so you can see that for the positives, these scores have shifted leftward to higher scores. And so for prediction, how we actually create the prediction out of these histograms, these fit curves, is that we calculate the score for each module, and then we go to that value in the probability distribution and compute the log likelihood ratio, which is just a sort of ratio between the probability density for the interacting curve and the probability density for the non-interacting curve. So we basically are just in log space dividing one curve by the other for that specific score. And then to combine together all of the modules, we just sum the log likelihood ratios for the modules that actually gave us a score. And the advantage of this is that if a module doesn't return a score, like string often doesn't, because if you don't have the pair in the string database, obviously you aren't going to get any features from that. So if it does return a score, it can just get added to the sum, but if it doesn't, we just leave it out of the sum. We don't have to do any sort of imputation or anything like that. And so in the actual paper for this program, we do a bunch of benchmarks, but this is what I think to be the most impactful and important one to discuss, which is an unbalanced benchmark where we took 100 protein pairs from the PDB, stuff that's straight from the Spring database, that we know is interacting because they come into physical contact in their crystal structures. And then we take those 100 true pairs, split them into 200 individual monomeric chains, and then pair those chains all by all with each other to create a pile of about 20,000 interactions that it needs to classify through. So essentially what we're trying to test it to do here is something that's akin to what you would actually be doing in a whole proteome classification case, that you would just be given the whole proteome, you would pair everything with everything and try to pull out the true positives out of the huge pile of decoys that you're creating. And so on this specific test, PEPI is outperforming all of the other programs with statistical significance. And interestingly, the only one that even came close to its performance is another homology-based PPI predictor that we have. PEPI is the blue curve here, and then we have this green curve, which is Sprint. That is a sequence-based protein-protein interaction predictor that does a little bit more sophisticated stuff with fragmenting the sequence, but we did benchmark against deep learning PPI predictors as well. So if you recall, when I said earlier, one of the things I wanted to solve was this issue of it being benchmarked in species-specific context. Well, this is a species agnostic problem now, and you can see that the deep learning methods are not performing as well as clearly their benchmarks indicated, or really what we would think deep learning should be attaining. And Piper particularly did awful, that the highest precision, it classified so many things as positive with a probability of 1, that if you let in all of those ones, the highest precision you can get is about like 0.2. So really there's a lot of room to improve in deep learning. And for now, homology-based approaches seem to still be outperforming the deep learning approaches in this field. And so one thing that I wanted to highlight is that I mentioned earlier that if you get good enough threading results, you can actually model what the interaction looks like. And so we have an example of that over on the right there, that those sort of jaggedy traces are the C alpha positions that we've modeled using Spring, and then the high transparency cartoon figures beneath it are actually the structural template that we use to create that specific dimeric structure. And so this program is done, published, ready for people to use. We have a web server that people can access. Over 400 pairs have been analyzed by it already. It was broken yesterday because somehow someone put in an empty file that caused the whole thing to not be happy, but I've fixed that now, so it should be ready to run for people who want to use it today. And so now let's move on to what we can actually do with this PPI modeling program, which is apply it to biological systems and see if we get any new insight on those biological systems. So in the manuscript for PEPI, we focused primarily on this SARS-CoV-2 and human interactome, where we took about 31 proteins of the SARS-CoV-2 virus, paired those against all of the 20,000 human proteins, and then classified each of those pairs and pulled out the things that were above a likelihood ratio of 1 or a log likelihood of 0. And so we got 403 interactions in total, most of those belonging to the spike. And the top rated interaction of this network was spike ACE2, which makes a lot of sense because that's probably the most well-studied interaction out of this entire data set. So we better have hit that. And some other interesting interactions involving spike include the S-furin interaction, which is unique to SARS-CoV-2 and not to SARS-CoV. There's an interesting feature in this network where you have this human protein that's attached to a lot of COVID proteins. That is SERPIN-G, which is a inhibitor. It's a protease inhibitor and involved in blood coagulation. And so it's interesting to see how it's interacting with so many different COVID proteins. And then finally, we wanted to benchmark how accurate this data set looked. And so we pulled out a gold standard data set of just stuff that had already been established in literature from Psychic. That's a database that basically queries a bunch of different databases all at once for high throughput experimental data. And we found that our data set overlapped with 72% with that gold standard database. So the network that I just presented is starting to get a little messy in 2D. That you can see there are some features of this network that are sort of difficult to visualize. The connections sort of get overlapping with each other. And so the underlying structure of that network is really hard to look at. And so not only that, but I also wanted to make the visualization of networks a lot more fun and accessible so that people who weren't necessarily scientists would get more excited about what a network could look like and sort of navigating the complexity of a network. So I developed this program Falcon, which is a VR program that visualizes networks in 3 dimensions as well as virtual reality, so you can actually navigate through this network like it's an actual physical object. So I'm going to do a demonstration of this program at its current state. It's still actively being developed. I think its current version number is 0.2.1. So it's definitely still in beta. And we're also currently working on getting this fully published. So features are going to be added to it. And it's definitely going to be changed in its interface a little bit. But I just wanted to do a demo of what it looks like right now. And it is available on this GitHub link. So if you want to use it for yourself, you might be willing to download it and give it a try. Especially if you have a VR headset, I'd be really, really thankful for anyone who is willing to test this on their machine in VR, because one of the reviewers said that he couldn't get it working, and I don't know how, because I tested it on other headsets and it worked fine for me, but I want I want someone to help me with that, so that if someone can't get it working, I want to know why. So let's do a demo. So you can see it sort of wobbling around. There's a light source there. So this is what it looks like. This is just sort of the screen that you get when you first open up the program. I don't know why my... Okay, that's weird. So my headset is misbehaving. This is not the program, I swear. This is the headset misbehaving. But so if we load the example network, which is the COVID data set that I just presented, we get presented with this network. It sort of looks messy just on first glance, but you can navigate around the network, look at it in 3 dimensions. We have all of these labels there by default, but we
Now,
can...
interestingly, if we were sitting in the middle and just crossed our eyes, you can see this in 3D. I'm gonna try it.
Oh, are you seeing? Those are both the same image,
Yeah,
so
so
that's
I'm
not
getting-
going to be
I'm getting
presented in
3D.
3D. Yeah, so
It's a little tough because the screens are spread out.
yeah. So
Yeah, I've got it now.
we have all these labels here. We can configure. Honestly, I think it's the bright light of the projector that's causing the headset cameras to freak out.
I believe it.
So
You want to step away from the projector?
yeah, let me see if I can.
You want to turn off the overheads? Let's do that too. That
Yeah,
might
that
help.
might help. We'll see. Well,
And I think that's good.
it's now freaking out because it's seeing a different room than what it saw before.
Well,
So-
now we know some of the problems that might have happened with your reviewer. You know?
Well, this isn't my software issue. This is a Oculus issue.
Yeah, right. That's
So-
the problem.
Anyway, so now we can configure the nodes. Let's actually. It detects these by the cameras, so it's not. There we go. Cancel.
You hang in there.
Yeah, let me see if I can move away from the projector. Let's go this way. I hope I don't smack anybody in the face if I do. Just figuring it out. Because I'm sure you can see movement on the screen, right? Because I can't see anything in here right now. But anyway, if this doesn't work, I'll just do mouse and keyboard for the next portions. Because there is in fact a mouse and keyboard segment that that allows people who don't have VR headsets to still use this technology. Yeah, it's really confusing. So I'm just gonna.
So what are you trying to show us?
So I'm just trying to show you the VR functionality of this program and the kind of stuff that this program can do.
And when you're looking inside your
I'm seeing
device,
Oculus default, whatever.
so it's not
So
working
it's-
inside your device.
Yeah, it's clearly the program is working. It's the headset that's not working. So let me just reboot the thing. Do it in mouse and keyboard.
But the big idea is that by being able to look at it in 3 dimensions this way and to interact with it,
Yeah,
that you see different features in the networks.
yeah, exactly.
And then what would those be?
So I'll highlight a few examples of those later, but just for this network, I just wanted to show the functionality of the program. So
Okay.
this is it in mouse and keyboard form.
Oh, good.
If you hit tab, you can configure the nodes, get rid of the node labels, or say you wanted to highlight just the Spike ACE2.
Okay.
We can color that specific color. And then I know just by magic of working with this data set forever that these labels are from PEPI itself. So we could rename them according to the actual protein names. So let's do that. Key name, select. So now we can see that we've loaded in all of these new names. Apply our changes. And so now everything has gotten rid of its labels except for Spike
Okay.
and ACE2. And those are the 2 colored nodes that we've put in this node group. Also, you can configure the edges. So you can change the color of those edges. Let's make them white just to make them very visible. And then also, specifically because I developed this with PEPI in mind, you can weight the edges. What I do here to weight the edges is by PEPI predictive confidence. That's probably fine. Ah, no, it's not fine. Maybe I opened the wrong file. Okay, I think I might have grabbed the wrong file for this, but you can, I'll show it on a different network that you can reweight these edges according to the predictive confidence that file that you get out from PEPI. So if we go back to presentation. So now let's talk a little bit more in depth about some of the data sets that I've been looking at with the PEPI program and some of the features that you might be able to pull out from the Falcon program that you weren't able to pull out in 2D.
Can I ask you a question about Falcon?
Yeah.
Are there limitations like in the number of nodes you could represent or
So
other
it's a,
types of things that like, what's the sort of scale and best use case versus sort of this?
yeah, so the limitations of the program and how many nodes you can render is entirely based on what computer you are running on. That I don't think the program is going to crash. It's going to be whether your computer runs out of resources first. And I haven't done those benchmarks yet. That's something that I'm planning to do for like a final paper on this program. I don't know what the upper limit is, but you'll definitely see with some of the networks that I'm rendering, there's also a time limitation of how long you're willing to wait for it to generate the layout for the network as well. That because it's 3D coordinates, it has to figure out where to position them in 3D. And for the networks I'll be looking at, that'll take like a minute or 2 before it fully generates the structure. So you'll see firsthand sort of where the upper limits are of this program. The 2 interaction data sets. Yeah.
Can I just piggyback on this question? So for the edges, are they like directed or not?
For now, they are non-directed. Directed network support is something that we plan on implementing in the future. That's like on the feature list for the next version.
Just another nice question. So for this network, are they based on structure touch or based on functional similarity?
So because it's from PEPI, it is structural touch. That's what the predictions are coming out of PEPI. So the 2 data sets that I've looked at in a collaboration with Vince Young's lab and Philip Andrews' lab, and used to be a collaboration with the Peter Fredolino lab, but now I'm part of Peter Fredolino lab, is trying to understand how probiotics and just sort of bacteria in general end up colonizing the gut. And so two probiotics that we were looking at is E. coli Nissl. It's very helpful that this is just a strain of a model organism. So this one's going to be pretty well studied. We know what a lot of proteins do, but definitely not all of them. And then L. rhamnosus GG, which is another probiotic, which is a gram-positive bacteria and is not a model organism. So it's a lot more poorly studied. We can get a lot more functional insight on the proteins of that network. And so to actually do this predictions, we applied a filtering procedure to the bacterial proteome so that we could focus primarily on the interactions which were most likely to be interesting in this network. The way that we did that is we took the bacterial proteome, we predicted with MetaGo the functional annotations, the Go terms that would be associated with those bacterial proteins. And then we filtered out any sequences that were associated with Go terms that seem to be associated with outer membrane location or pilus assembly, just these really definite Go terms that are associated with something that's going to be on the outside of the cell. Or if it's also involved in secretion, that's also something that we want to grab. But to really grab the things that are likely to be secreted, we also filtered out proteins that, or I shouldn't say filtered out, I should say isolated proteins that were predicted to have a signal peptide, which implies that they are going to be part of some sort of secretion system and be excreted from the bacterial cells. And so from these filtered bacterial proteomes, we then, it filtered down the proteomes to about 10% of their original size. And then we paired those with the human proteome to do our PEPI analysis of all of these interactions against the human proteome. So if you'll note, there is no intracellular interactions going on in this network. It's only inter-species interactions that we're focusing on. So this is what the network structure looks like for E. coli Nissl. You can see it's way nastier than the one I gave you for SARS-CoV-2. You can see there are huge hairballs here that are really hard to visualize. And this is about 2,200 interactions that we've isolated in this network, which is only about 0.02% of the actual interactions we had to calculate through. So it's good that this thing goes fast because that's a huge pile of protein interactions and a huge pile of data points that we have to assess with this algorithm. So I also have a distribution of what proteins were getting the highest, what bacterial proteins were getting the highest amount of interactions. The top two are not very interesting because CIBC hit a GPCR. One of the known issues with PEPI is that because it's based in homology, if you hit one protein in a family, it tends to also hit proteins of that same family. And because GPCRs are so structurally homologous with each other, when it hit one GPCR, it hit all of the GPCRs that we know about. And that's a lot of them. And the second one is not as interesting because this is just an artifact of really the PDB rather than an issue with my program that MAL-E is the bacterial protein, and that is commonly used in protein purification as a purification tag that people will use this maltodextrin binding domain to bind to a maltodextrin column to purify their protein. And then when they crystallize, they just leave it on. And so some of those interactions are almost certainly going to be actually biologically relevant, or otherwise they wouldn't get hit in the first place. But it's hard to tell which ones are real and which ones are not real by PEPI alone. So we sort of have to rely on other methods to potentially classify which ones are going to be real and which ones are not. However, the next one on that list is Ecotin, which is similar to SERPIN-G. It's also a protease inhibitor, so it's really interesting to see the kinds of interactions that we're getting out of that. So let's isolate a few features of what this network looks like in PEPI. And actually, while I'm discussing this, I'm going to open it in Falcon so that it has time to figure out the layout, because this is the biggest network that I'm going to be looking at today. It's, like I said, it's about 2,200 interactions. So it's going to need some time to figure out the layout for that. But once we finally get the actual layout, this is what it's going to be looking like. That this is the whole network all at a glance, which may look messy just because we're taking a 3D object and rendering it in 2D. There are definitely some parts of it that are still hairballs because we can't go higher than 3 dimensions, and the actual underlying complexity of that network is higher than we can visualize. But there are definitely some features that have been made a mess in 2D that we're going to pull out in 3D. So one thing that I wanted to highlight here in the C part of this figure is actually the edge weighting scheme. That this cluster is ecotin, and this cluster is some other protein that has actually a non-normal gene name. It's like an ORF name rather than an actual gene name. So I don't actually know what the function is of that protein. But you can see that the interactions with ecotin are on average way higher confidence than the ones for this other protein cluster. And actually this specific pair of clusters is really interesting because in the 2D representation of this network, they put these two clusters completely across from each other. You can see that there's this one edge that sort of spans all the way across the network to connect those two. So the fact that they're able to be close to each other in 3D space just sort of highlights the utility of using 3D space. Another feature that I'll be looking at is this feature right here, which involves a lot of the OMP proteins. You can't really read those labels in this this version of the figure, but there's the OMP protein, and then there's a DEG-P protein that branches off into another cluster. In 2D, this just looks like garbage, it's hairball garbage, but once we actually look at this in 3D, it's going to become more clear that it's sort of a pseudo-cylindrical shape, that it does have some things that hang off of it, like this DEG-P star-shaped structure, and there's definitely some stuff up here that that hangs off the top of it, but the underlying structure of this becomes a lot more clear, whereas in here, it just looks like a mess. So let's see if it is figured out. Visualization, it looks like it has. So let's get rid of these labels because that will affect how much rendering we've got.
Close that window out.
Yeah. So here's what the entire network looks like. Let me sit back down so that I can navigate. So this is what the entire network looks like just when you first load it up. Here's that feature I was talking about earlier, where it just looked like a mess before, but now you can see that it sort of forms this pseudo-cylindrical shape. And if you wanted to look at the actual connections between the nodes, you could just wobble from side to side and see more clearly which nodes are connected, which ones are not connected. And then you can see this, there's this sort of pseudo-cylindrical shape, and then there's the star shape that I mentioned before with DEG-P. And then here's the feature just sort of hanging off the top. You could see that in the actual figures that I showed, there were a couple things that I did to spruce it up, like coloring versus the bacterial proteins and the human proteins, as well as I could also do the edge weighting. I know what the problem was with the edge weighting last time, that I renamed the nodes before I did the edge weighting stuff. So if we do the edge weighting now, works completely. We change the edge color again to white. Apply these changes. Now we've got the edge weighting. So you can see that this DEG-P star feature is very highly confident that all these interactions are confident with each other. Some of these bits that are hanging off the pseudo-cylindrical structure are less confident, so they might not be real interactions.
So is that
Yeah, both of them are red right now. In the figure that I showed, the two-dimensional projection of the figure, I've colored them so that they're blue human, red bacterial. But I would have to go and assign by hand all of these nodes to the two individual groups. And that's no different from like a site escape. Site escape also forces you to assign these nodes by hand. But yeah, so this is what the interaction network looks like. Looking for the Ecotin cluster. There it is. So there's the Ecotin cluster. You can see that it has a lot of connections with this cluster here. I'm not sure which that is, but and then there's the other protein that was far apart in the two-dimensional projection, but now is close together and visible in the three-dimensional projection. And so this is the L-rhamnosus LGG network. It has similar features to the E. coli missile network, but the degree distribution, the interactions that are spread across the different bacterial proteins is more even because we don't have those weird false positive peaks. And the top, I'm trying to remember exactly. I believe it's a cytochrome protein, but I might not have that right. So but you can see by the labels on that node degree distribution that these proteins are not very well characterized, and many of them do not have like normal canonical names. These are like open reading frame names that have been assigned to them. So there's a lot to learn from this network. And I'm just going to be highlighting some features of the network. So we have much more clearly another pseudocylindrical feature like we did with E. coli missile, but this one's a lot cleaner to look at. And you can see that it sort of has this alternating pattern in the layers of this pseudocylinder of human and then bacterial and then human and a bacterial. And you can actually sort of get a glimpse of that in the 2D representation, but the connections between them are completely muddled with each other, and it's really hard to figure out what's going on. So let me actually load this network into Falcon so that we can take a look at it. Let's reinitialize the program. Now this one I'll sit through just to show you that it does still take a while to load in this network, but it will be done in like a minute or so. I believe this amount of interactions is like over 1,000, so it's not an insignificant size of network, but it still does take a little bit to figure out what it's looking at. So there's what the network looks like when you first load it in. There is definitely some cool looking features like that would not look good in 2D, but it's like this star structure that's really cool to look at.
Eric, are you getting some kind of insights into some of these different topological forms, what they might signify in terms of connectivity and function?
Yeah, so the actual topology, I haven't really looked at that much in depth. The kind of things that I've been writing about in my thesis on this stuff is like specific interactions of interest, that specific edges that might look interesting as part of this network, but
or connections between the various networks. You know, there might be
Yeah.
opportunities there.
yeah, definitely. So there's a lot of analysis one could do with these networks that I just only barely scratched the surface of. So hopefully someone who's much more of a systems biologist than I am would get a lot more insight out of this sort of visualization than I was. So let me see if I can find that pseudocylindrical form that I highlighted earlier.
But you definitely get more
It's right here.
insight into the stellated forms and what's the hub and what are the nodes, which would likely be obfuscated with the planar view.
Yeah, exactly. So the connections here, even though you sort of see the layered pattern in both 2D and 3D, you can much more easily look at what is connected to what if you can wander around it in 3D.
Rick, if you have this network and you wanted to actually see what's going on, is there a way that you could share the information about the Like, let's say you click on an edge, like the supporting prediction or evidence that goes into it.
Yeah, that's definitely something we've considered for future features. That's just going to be pretty difficult to implement and also specializes it to a specific field. So the thing that's one of the advantages of this program is that even though we're using it for PPIs, there's no reason why you have to do a PPI.
Exactly.
That you could put any kind, you could put non-biological networks into this thing and it would still be able to visualize it. So we definitely want to maybe develop like plugins or something. I don't know. We're sort of tossing around ideas of how we could integrate that sort of information in, because like ideas that we've had before is like grabbing the edge and then seeing what the structure looks like. That would be really cool, but that also means that we are building it explicitly as a companion to PEPI and not just as sort of this generalized network visualizer.
What does a tooltip look like in 3D? Like, could you have a little bubble that comes up with like kind of a,
Yeah, so that
like a web page? Or
usually what you get is what I briefly sort of touched on in VR was you get like a 2D projection that turns into an object in space. And so if stuff is overlapping with it, it'll cover the actual tooltip. And that was a problem that a bug that I didn't anticipate that reared its ugly head when I tried to bring this into the public, was that there was actually a node covering the close button for the actual window. So I had to figure out an alternate way to close that menu.
Could you have like a 3D tooltip? Like, could you bring up the protein interaction?
Yeah, I definitely could. That's a little bit more complicated 3D coding than I generally know how to do, but that's definitely something I could do in the future. Yeah.
So I've been doing the hybrid model. Your visuals are fantastic.
Thank you.
What I'm curious is, do you have some general strategy of selecting the networks that you want to analyze? You know, there's several bacterial models, but what are the properties that you know in advance make it a good choice for applying your methods?
So hypothetically, the method could be applied anywhere. These
You've chosen some.
Yeah, the ones that we've chosen, I mean, specifically SARS-CoV-2, that was because that was obviously a hot button issue at the time, that it's really urgent that we figure out how that was working, because at the point we had started doing that, there wasn't even a vaccine yet. So having that sort of interaction information is crucial to.
Did you get a lot of citations with that?
Not quite, because it got lumped in with the PEPI paper. It was not its own paper. The other 2 bacterial species that we were investigating, I wasn't involved on the grant writing process for that, so I don't know the exact reasons, but I know the specific ones that they picked. They wanted to pick a gram-negative and a gram-positive so that the sort of analysis we did could be generalized to most bacteria in general. They wanted to pick probiotics so that it had a positive health impact as well. And then finally, of course, E. coli, we wanted to study strains of E. coli because there was a lot there that we already knew existed and should be able to retrieve. Basically, that was the main justification. I don't know exactly why LGG on the gram-positive side of things, but that's sort of the justification behind why we wanted to pick all those specific bacteria.
Well, that's very helpful. I'd just like to comment to everybody that when you do a methods development and applications, and you pick some good examples for demonstrating its usefulness, it's good to specify in advance what the properties are of the systems you think would be beneficially analyzed, and then to examine those in light of what you've learned in performing analysis.
Right. So in summary, the stuff that I've talked about here is the PEPI program, which performs species agnostic direct PPI prediction. And then what I found from that program is that homology-based approaches are outperforming deep learning programs, specifically because of that species-specific constraint. And I want to say overfitting to those specific species, but maybe that's a little bit overreaching. And then we use PEPI to predict PPI networks in several cross-species biological systems, and then visualize those in Falcon. So Falcon will continue to be developed. I've mentioned a few of the features that we've considered putting in like the directed network support, and potentially just being a little bit more informative when it's invoking its layout, that like we give it a loading bar for how many iterations it's performed versus how long we're going to give it. And then also one thing that I didn't highlight about Falcon that is actually an important feature about it is that its ability to load multiple states of the network that you can switch between. Right now, it's not very useful because it requires that the node set stay the same, but we're working on developing a way to break that sort of dependence on the node set staying constant so that it's a lot more useful, so that if we say, oh, we have this this specific set of connectivities and this specific set of connectivities in different conditions, you can visualize those and swap between them back and forth. And then finally, PEPI is also being improved through the addition of interchain coevolution. And I've run out of time to be able to talk about that, but I'd be happy to talk with anybody afterward if they're interested in the kind of stuff I'm doing on that front. So there's definitely people I'd like to thank: Jake Schwartz, Yang Li. Those are sort of the people who've been helping me most with getting things done on this program, as well as some of the deep learning stuff that I'm doing in the future. And then plenty of other people in the Fred Alina lab, my FACES committee, many of which who are in attendance today, and funding sources. So thank you all for coming, and I'm sorry the VR didn't work out as well as I hoped it would, but you at least got to see that it can do VR. So
Yeah. It's a bleeding edge, right? Wow, very nice.
There is a very lengthy question on chat. I'm happy to read to you.
Holy.
Oh.
Looks so long here.
PCR. Maybe we can invite him to talk.
yeah, so I think the general bit of the question is asking whether knowing the species would help us filter out which GPCR structures were more likely to be actually truthfully interacting and which ones were not. So if you're saying the species of the structural templates, maybe, but I think even if we had just one structural template of the GPCRs, the fact that all of the human GPCRs are so homologous to that one structural template, it wouldn't help the filtering process. All of them would be hitting that same one template. And on the actual like input sequence side of things, it actually is species filtered because we're only pairing it against the human proteome anyway.
But
So
the GPCRs are in multiple different family, branches of the tree.
right, but in fold, they're all pretty homologous.
Okay.
They all have that same transmembrane helix structure that is conserved. In terms of the extracellular domains, they might be a little divergent, but that strength of homology in that transmembrane domain definitely causes you to hit it time and time again if you hit one GPCR.
Anyway, to see if they're happy with that, Marcy.
Is the screen? Oh yeah, I have chat up right now. So
Yeah, it seems like that did answer the question.
Yeah, so you
Thank
might
you.
want to take the chat off. There we go.
Are there any other questions in the room or online?
For Falcon, it looks like the graphics are from the late 1980s. I'm
Maybe.
curious if you if that's a style choice or a limitation of the system, or like if there's, it could be for clarity, but it also
when you say
Would you
graphics, you
include-
mean the UI sort of stuff?
like there's no shading, there's no like.
Oh, there is definitely like lighting
Okay.
to it, like there's reflections on the balls that move, and there's also a reflection on the floor. So there is a lighting system. It's just neon, which I guess in that case, yes, was a stylistic choice.
Okay, got it.
But
Yeah, they're
So a
self,
lot of
they're
this-
self-luminous. I mean, it isn't a perspective,
Yeah.
a lighting source.
No, there is a lighting source. So it's,
It's a very bright sun, very dark.
I was wondering if you guys had considered possibly adding like borders to the edges, 2D borders, just because like, I think for some of those really interconnected things, I mean, maybe it's more clear in 3D space, like in the, when you're using VR, but some of them kind of, a lot of them kind of blend together, a lot of lines kind of blend together.
yeah, that's gonna be a little difficult because the edges themselves are like sprites, so they are borders already. So you can't add borders. There might be some way that I could do something like ambient occlusion, which I don't know if you're familiar with that at all, but
Sort of, yeah.
the idea is just that every object sort of has a black space around it so that it looks like every object is sort of stuck out from its background, which that might help. I wonder if I can implement that easily, but that's the actual graphics side of things is a little bit over my head. This was my first foray into Unity. This is developed in the Unity game engine. So a
That's
lot of,
a very successful company.
yeah. So. Yeah, I agree that the edges kind of do get a little bit overlaid. I wonder if there's something that I could do with like the in to out gradient of the edge. So I could make it like a softer edge or something like that. So they weren't quite so easily overlapping. Or maybe do something like how in Primal, when something's farther back, it's more transparent. Do that sort of a thing.
Great.
You have a date for your thesis.
Yep.
That's all right.
November 1st.
Number one.
Which is like that far away. That's
He's ready.